Welcome to Revolution Against Evolution. I'm your host, Doug Sharp.
Your co-host, Rich Gear, here with you. And once again, we want to talk a little bit about our Arizona trip, Doug, and specifically a very gracious host, Kevin Anderson, who helped really blew my mind away talking about a lot of neat stuff over at the Van Andel Research Center or whatever. What do they call it? Actually, what do they?
It's the Creation Research Society Van Andel Research Center or something.
That's too many words for me, you know. But anyway, Kevin really shared some really amazing things. You've been getting in on some of this stuff because this is one of Doug's big things, the genome and the biology and all that kind of stuff. But when he started talking about some of this stuff, there's all kinds of stuff they do there. But that thing really floored me.
Yeah, Kevin, we just got him started talking, and he just took away with this. I really don't have to explain too much about what he's going to talk about because he's just so animated and exciting. A little bit about the research center. It's just north of Prescott, Arizona.
Yep.
And just north of a town called Chino Valley, and it's sort of out in the boondocks. About halfway before you get to Ash Fork, and you can see in the distance the Coconino Plateau where it actually raises up. You can see cliffs in the background where that raises up and the muggy in the rim, and there's and it's just a little bit west of Sedona, and so you have to. In order to get there, you have to go sort of a side trip.
You really have to know where you're going and want to get there, because otherwise you'll never find this place. I mean, it's not that hard to find, but it's off the beaten track. It's true.
But they have electron microscope there, and they have a lot of equipment and research equipment, and several scientists spend quite a bit of their time at the research center. And so we were in the back. It was actually a garage or something like that. It's a storage, where they have all the publications that they send out for the Creation Research Society. And while we were in there, some of them were starting to ask Kevin some questions. We had a real big group from the Arizona group, and Kevin just got started, and it was hard to keep him held down. He was just 2 feet
And guess what? We're going to do the same thing. We're going to let Kevin just go for it.
It used to be a gene was identified as a stretch of the DNA that was transcribed and the transcript was translated. And that was your classic definition of a gene. It doesn't work anymore because it's far too complicated than that. There's still segments where the concept of a gene applies. But by and large, throughout the chromosome now, the concept of what is a gene really doesn't fit anymore. So it's an outdated term.
The four-dimensional years, they're not holding together like you say.
Well, it's not a matter of holding together. It's a matter of just strictly the concept of a region that's transcribed to be translated. That only applies to a very small segment of the DNA anymore. The rest of the information content of the DNA does not fit that pattern. And even the gene itself is not readily identifiable anymore because there's so much overlap. There's so much buried gene inside gene, reading frame inside reading frame. You got all the splicing that goes on in the transcripts. I mean, some of this has been known for years. Some of it's only been coming out in the last decade or so.
Does this
but
go
when you put it all together, we no longer now have a very clear idea of what we would call a gene. A gene no longer really fits a very easy definition. There's just too much to it. Yeah. When I teach genetics, I still teach gene. But what I have to say is I have to put in the caveat, you got to recognize that we don't know what a gene is anymore. So I have to fall back on the classic definition. This is what a gene is, so I can communicate to you as students what we're talking about. But once we get past that introductory level, we now abandon the classic ideas because they don't fit anymore. And now we're wading into this whole nuance of areas we don't know what's going on. We don't know how to define this, how to describe this. So we don't have the words to use.
I like your passion.
Kevin, does this go back to where you told me we were at that mega conference, and you said that the DNA was getting where we were in this one room.
Yeah.
And then you guys finally found this door, and you're peeking around.
It's like you found King Tut's tomb.
All right.
And you thought, wow, okay, look at this. It's got all this gold. Look how lavish it is. This is wonderful. and you're exploring through it, and you're cataloging everything, and you're understanding what's here and what's not there and such. And you think, okay, we've got it all cataloged, we're getting where we understand it, we're getting where we have most of it identified. And suddenly then somebody notices a little crack in the wall over on the one corner, and they kind of push on it a little bit.
Okay.
And suddenly you realize there's this whole hallway. And you start traipsing down the hallway, and then all of a sudden you enter into a chamber that is far up as you can see, and as far down as you can see, and as far over as you can see is nothing but magnificent treasures. And you recognize that was just the teeny little antechamber.
That was the teaser.
Yeah, that was the teaser. This is what it's all about. This is lifetimes worth of work to do, and that's where we are genetically. We thought we were getting a handle on what we had.
Right, they mapped the human genome.
And we were,
what, 10,
we
15
were
years?
just in the little antechamber. And now, as we're understanding more and more, recognize no, we're now moving into that huge chamber. And the genome, as far as we can see up, and as far as we can see over, and as far as we can see down, is so beyond our comprehension that it'll be lifetimes understanding
Wow.
what is going on in this thing.
So is
And
this
accidents
So
don't
all
put
of a
that
sudden,
together.
all of a sudden, what we thought we knew, we now throw out because that's grade school.
That's the sixth grade thing.
Yeah.
Ventnor said that he was interviewed and he said, well, now that you know the sequence of DNA, what medical advances do you see? He said, none. Because we just know the sequence now. We don't know hardly anything else about it. But are there 20,000 or 30,000 human genes still? Is they
See,
still
that's
use
back to the archaic definition of
Okay,
gene.
so-
Yeah.
It doesn't work anymore.
Yeah, it doesn't work anymore,
So-
exactly. And that's
But how
where
did they
polycystronic
isolate-
comes in as well. See,
What does
because
that
they
define
were
that term polyhystronic? What does that mean?
Okay, and I talk about that in my video. Polycystronic is there was a time, and this is what I was taught.
Okay.
I mean, it took me the longest time to accept this. John Sanford was the first one that told me about this. I made him repeat it 5 times because I didn't believe it. And he and some others finally produced the paper and said, here it is. And I sat and read it. DNA double-stranded, 2 strands.
Right.
So the concept was that one of the strands is what we call the sense strand. And that would be, it's the strand where the gene is. It's the strand that contains genetic information. The other strand in that region, the other strand would then just be the replication template.
Right. Okay.
Because, let's face it, the sequence of each strand is complementary. They're not independent of each other. So what the sequence of one strand is really defines the sequence of the other. Okay, so if you're going to have information on one strand, you're not going to have information on the other strand.
But you do.
But you do.
Yeah.
And that's what polycystronic is this region where you are transcribing to a gene, using the classic definition of a gene, okay? This region where you're transcribing, instead of its complementary region being just the replication template to make the double strand, it also can be transcribed. And I made him repeat that over and over because to me, that was so mind-blowing. Not only as a creationist, it was so mind-blowing, I can't even understand how an evolutionist would not just get up and run away. Because now all of a sudden you're introducing concepts that Darwinism can't even comprehend, let alone offer explanations
You're
for.
getting way into it,
Oh,
way too deep.
oh,
and they only
way
like
too
the
deep.
external
Way
fluff.
too deep.
And
We're
telling
not talking
people-
about monkeys typewriting the Encyclopedia Britannica. We're talking about monkeys typewriting something that is beyond what can even be read in classic structure of left, right, top to bottom. We're talking about monkeys typewriting something that, as I use the analogy, it's like taking a 1,500-page book of War and Peace and putting it on 15 pages. But it's not 15 pages of small print. It's 15 pages typed in such a way that you read it left, right, top to bottom. Turn it over, left, right, top to bottom. Turn it again, left, right, top to bottom. Then you read it bottom to top. Turn it over, bottom to top. Then you fold a page and read it again. Then you fold a page and read it again. Then you fold a page and read it again 100 times.
Flip it backwards.
And then you go to the next page. And it's not like each page is independent of the other. It's all one continuous story. So there's that continuity. Can monkeys type that? We can't even type that.
Yeah.
Yeah.
Well, no wonder we think we come
that's
from
what,
monkeys.
Yeah, that's what the Darwinists are faced with now. It's not a matter of monkeys typing Encyclopedia Britannica. It's monkeys typing something that we can't even type. We can't even comprehend how to type it. And I challenge audiences when I present this, could you read that? No.
And then
Could
you throw
type
time
that?
into it?
Yeah, could you type that? No. Could you read that? Of course not. But guess what? it has been typed. And it is being read.
Wow.
Every second of every day in your cells. Yeah, and you throw that fourth dimension, the dimensionality of time in it, so the DNA is actually a four-dimensional structure because every second it's changing its shape, its form. So have monkeys type an Encyclopedia Britannica written in that way that changes every moment.
Yeah, I have the typewriter there
Yeah.
and have it moving around on
Yeah.
the monkeys. Now type something.
So that's what I mean by the polysystronic, is that you're now talking about both strands
Okay.
are information laden in the same area.
And then you
So
can
if
read
a
them.
mutation occurred to one strand, it occurs to the other strand as well. So even if it's a so-called beneficial mutation that somehow now gives this reptile the ability of having wings and flying, amazing mutation, it's got a mutation in the other strand that's going to have a effect as well. Is that effect going to be benign? Is it going to be neutral? Is it going to somehow also be positive? Well, we know enough about mutations to know that's not what happens. What happens is even if you introduce a mutation on one strand that somehow has some positive benefit, you're going to have another strand of mutation that's going to have a very much counteractive production.
Wow.
And so now you can't change the one strand without the other. It's like if I'm typing a page, and every time I type that page, I affect 100 other pages. How can I possibly make a one-word change on one page and keep the book the same? Because 100 other pages have now been changed as well. So that's what polysystronic does, is it creates that tight continuity that does not allow for changes. In fact, the genome is designed to be very rigid, to be very stiff, resisting changes.
Read only. Can the double strand be read at all in any way?
You mean both strands at the same time?
Both strands together. And
I'm not
reading
aware.
The contours
I'm not
of the
aware
double
that
strand.
that the strands are read simultaneously.
Okay.
They're read always individually. The polymerases read
So
each
they
strand
always have to
individually.
be read
Yeah.
when they're separated.
Right. I'm not aware of any. That doesn't mean there isn't.
Right.
But I'm not aware
Wow.
of any mechanism that
And the
reads them as a
unraveling has to
collective.
be
Yeah.
done by enzymes.
The
right?
unraveling is done by enzymes, and you got to wind them back up very
Yeah,
quickly.
wind it up, then-
Yeah, and that's part of that constant changing of the size and shape, is that raveling and unraveling. So you got that chromosome very tightly packed. You need to get to a certain gene, so that means you got to yank out that segment of the gene. So it's got to go out. You transcribe it very quickly. It goes back in again, and you pull out another save. It goes out. You transcribe it. It goes back in. So if you want to think of it, this is constantly going on all the time.
And they all know exactly where they got to
And
be.
they all know exactly what's going on.
Good
Because
night.
proteins
Where they're
are
going.
all
Yeah,
are all
all at
directed.
one time.
Yeah. See, DNA itself is inert in that sense. It's like the paper. The proteins are the ones turning the pages. The proteins are the ones reading it. Okay, so the proteins know how to read it. They're the ones that know how to which segment to grab, to pull out, to transcribe, to then let it bounce back in to pull out the other one. But how do the proteins get their information to know how to do that? Well, it's what's on the DNA. So the DNA contains the information to tell the protein how to read the DNA. So it becomes a
Did you say
self-written.
that the information is then translated to the protein? Is the protein carrying information now, or is the
Oh, the protein
original-
has to be carrying information. I mean,
Okay.
the protein is a product. If you
Right,
can say
well,
the protein's
protein-
a product information, but it'd be a little bit like if I'm reading a page out loud. Is my voice conveying information? Of course it is. Where's the information coming from the page on the book? So that's the DNA. The reading out loud, you could say, is more analogous to the protein, or maybe more analogous would be if I'm reading the page and it's telling me something to do. Go over here, walk over here, sit over here. So my action then is dictated by the information on the page, but my action conveys with it information because of what I'm doing. I go pick this up. I go move this over here. So to me, and I've had this position several, challenge, if you will, and not all creationists agree with this, but to me, you have genetic information and biological information. They're not the same thing. Biological information contains genetic information, but genetic information is a subset of all biological information because proteins are information laden themselves. Proteins are not genetic information, they're protein information. So within the cell, you have enormous amounts of information going on directed by the information in the DNA, but it's now separate from as well, because the action the protein does is not the action of the protein. Or excuse me, is not the action of the DNA. The DNA may have contained the information to direct the protein what to do, but the DNA now becomes the protein now becomes a separate entity. And what it does, and the information it contains, knowing what to bind with, knowing when to bind, when not to bind, that's its own information structure in protein form, if you will.
In some ways, it seems like the molecules know what they're doing.
And more than in
Think
some
of that
ways, it's
like that way.
amazing. They know what they're doing. When I
As long as
teach
it's
my
delicate.
students, I say that enzymes have capacities up to 10,000 reactions a second. If you think about 10,000 reactions a second. So when you're looking at some of these pathways, of course, when they're studying the pathways, they're thinking in terms of, okay, this step happens, and this step happens, this step, and it's really, and you're done. You're done. You know, it's faster than you can blink that they go through the whole process, and you start over again.
This is
That's
why
how
the Bible
fast it's
says
going
we're
on.
fearfully and wonderfully
Yeah.
made. Because God figured out there's no sense trying to explain that one to us.
Is it possible?
No, the evolution would be seriously stranded. DNA stranded.
Yeah.
Genetic mistake without screwing everything up at the same time?
Presumably, the simple, you know, like cystic fibrosis is really just a point mutation. It would be theoretically possible to go in and replace that point mutation with what's the original base that's supposed to be there, and you cure cystic fibrosis by gene therapy.
So are they making strides along the avenue that you just outlined to us, using that, considering that as they're attempting
I mean,
to...
there's certainly being work done. I think the limitation in some ways is how to introduce it in a vehicle that actually will, you know, we may know this is where the mutation is, this is what it would take to fix it,
Yeah.
but the actual vehicle of trying to get it into your cells to
Yeah.
fix it, you know, there's still a gap there.
Almost sounds like doing a lobotomy in the past, what they used to think what they were doing and going in and sticking something in your brain. It sounds like you have danger of doing that when you are attempting to change.
How do they go about knocking out a gene? Like in mice, they knock out certain genes and see what happens.
Well, a lot of that's, of course, done at the embryological level. You go in while the mice is in the embryo state, and you can introduce various vehicles, if you will, that will turn off or disrupt certain genes. So as the mouse develops embryologically, it doesn't have that gene, or it's going to lose that gene. So, and that's the same thing with human. The idea is we're going to try to cure cystic fibrosis. The idea would be to try to do it in the embryo state, so that the individual is born without cystic fibrosis, not to come back when they're 5 years of age and try to. Doesn't mean it couldn't be done, but the idea is to try to catch it, before they're born.
Are you saying that some of the code in the DNA for one characteristic may overlap the code for another characteristic, or it could be read backwards?
Viruses, we see that all the time, especially bacteriophage. We've known that for years in bacteriophage. It's more slowly being recognized in the actual cellular DNA. But yeah, very much so. It's polycistronic, it's overlaps, it's reading frames being buried inside reading frames. So that it's again, you would have, by analogy, you'd have the words on the page, you'd read them one way. But now it'd be, all right, you start halfway through and read, it and change the spacing between the words, and it's a different page. Still part of the same continuum of the story, but it's a different page. But it's based upon the same words you had, the same letters you had on that page. It's now that you're reading them differently. You change the spaces. So, an A from this word now moves over and becomes the last letter of the previous word. And so suddenly, but it's not gibberish. It's not like it's not like that these are now, misspelled words. They're true, complete words, complete sentences, complete paragraphs, perfect English, if you will. Story goes on.
So it's the Bible code of DNA, right?
Yeah, in that sense, right. In that sense, it is.
This one is a real code.
So this is a real code,
it's
right?
real.
And we can see the effects of it all the time. Yeah.
Are they learning, so they're learning what some of the stuff does, or are they?
Oh yeah, that's been going on for years. That you slowly are learning what it does, what gene does which. That's where even evolutionists in the know anyway are abandoning the whole idea of junk DNA, because they recognize that, and to some extent, in fairness, the actual molecular people were never very strong behind the junk DNA. That was more a popularized.
Oh, there's a media thing. They jumped on that,
but a lot of non-molecular evolutionists jumped on it as well.
Yeah, but to me,
And
they use
promoted
that
it.
argument not because they understood, but because they didn't understand.
Well, that's true. It was an argument of ignorance. It was also an argument of convenience.
Exactly.
Because it's like, this obviously is something the creationists can't explain, they would claim,
Yeah.
and therefore it must be true, which is always kind of interesting, but
Yeah.
also it makes a good argument.
Yeah,
Just
well,
a pejorative.
When I first saw the junk DNA
Exactly what
argument,
we would expect, yeah.
it reminded me of the vestigial argument, only knocked
It
down
was.
a notch.
It's the
And it just
vestigial
says
argument
this is
at
gonna
the
go the
genetic
same
level.
place.
Yeah, it's a vestigial
And-
argument, same level. It's leftover. It's your leftover fish genes.
Yeah,
you know,
it's just-
your leftover reptile genes. Yeah,
Reminded me,
that's your
what,
gene
you know,
for
these are-
gills. Yeah.
Well, like the Rady experiments disprove spontaneous generation from a macrobio. Then Pasteur goes down to this. So then they go down to the subatomic level, and that's what the DNA. Okay, we did. Well, this vestigial that doesn't work. So let's go down one more. This is not going
Yeah.
to work. They're going to try to go down to the. They're going to go down farther.
And
trying to figure out.
the 1% myth. 1% difference between us and chimpanzees.
Yeah, that.
Yeah.
Yeah.
Yeah,
Baloney.
and
Yeah,
that,
but it's
that was
bigger
another
than
one
that.
that was another one that
Oh,
was
yeah.
promoted, not quite so much by the actual evolutionary geneticists as it was just popularized by the press and by Discovery Channel
All
and
those
by your
things.
what we say your less informed evolutionists that jumped on that again as excellent illustration of common descent.
Yeah,
And
that
that's
was
what we would expect if our closest evolutionary neighbor is chimpanzee. We would expect our DNA to be very close to them. Well, of course, creationists don't deny that anatomically we have a lot of features similar to chimpanzee
We have the
versus
same
an elephant or a
biochemistry.
horse, and so it's a lot of the same biochemistry, and therefore we would expect that there would be a greater similarity between us and the chimpanzee than would be between us and a horse. I mean that doesn't in any way contradict the creation model. But the whole idea of the 1% was pushed to try to make us so close. I mean, a few years ago, the Spanish Parliament, was promoting giving certain rights to chimpanzees and other simians
There's
based
still
upon the
people trying to do
1%
that.
argument.
We just need to
So you
elect them to
can't-
Congress.
So you can't say
What I
that these
always
things
was-
don't have impact.
What I was amazed about that was though they do the 1% for the chimpanzee, and then they show how far apart a Neanderthal and a human being were. I mean, it's like, wait a minute, a Neanderthal's got to be close. Why
Yeah.
they're so far apart, and yet you're saying this is so close.
You
and
know,
yet they're farther
interestingly,
away.
the mitochondrial DNA
Oh.
in Neanderthal is actually further from chimpanzee than contemporary human. So we're actually less evolved than they are, mitochondrial
Yeah.
DNA wise. But the whole thing with the 1%, and in my video I talk about it some, that even evolutionists are saying 6%. The
Yeah.
6% is still a crunch. 6% is still being very, very, very generous.
Yeah.
I quote one evolutionist who was a real big proponent of the 1% for many years, is just saying, okay, the truth of the matter is we just don't know. It's really more a statement of our particular view of ourselves than it is of the actual data,
Right.
which I find is interesting when evolutionists admit that they're really saying that evolution is really a promoting of the idea of how we view ourselves. In other words, our worldview.
So how can we claim to know anything about anything then?
Well, I don't know, because obviously the way our brain apparently developed, how would we know? The brain developing the way it's supposed to do by evolution, how can you trust any of your thinking?
Yeah, that's right.
It's a
Well,
sign-
they use
sign of
irrational
intelligence.
processes.
Yeah.
So how do you get rational
Yeah,
thinking from
yeah.
irrational
Yeah.
processes?
It's a little bit like
live
in
in
the
a
cell we
postmodern
evolved a
world,
repair
Marcel.
mechanism.
Yeah,
We
you
repaired the
bet.
DNA mutations, of which were responsible for making the repair mechanism to begin with.
Knuckle draggers for
And that obviously
Well, you
flies
get that.
apart very quickly when you look at it.
So as of right now, what percentage of the DNA codes for protein of the human genome?
Well, I think the
5%
estimate is that we make about 100,000 different proteins.
Right?
It's still probably maybe no more than 10% that we actually would identify.
As
But again, you got to recognize so much of that other percent is
Is
so heavily
controlling.
involved in
yeah,
Yeah.
that
and
10%.
gene regulation.
Yeah.
See, my chemistry professor once told me that the more he learned, the more he realized how ignorant he was.
Right,
And that's a
right.
sign of intelligence,
Yeah, yeah.
admitting your
Yeah,
ignorance.
the more you know you don't know.
Right.
And that's what the genome has done to us, is as geneticists, the more that we've learned genetically, the more we now know we don't know. And so now we're standing in this room as far as we can see, and as far as we can see, it's full of treasures,
Wow.
and we're just now beginning to try to disseminate through that.
This
It's
goes
like,
right
okay,
off the
yeah,
charts,
Yeah,
doesn't it?
yeah.
So- Goes right
like
off the charts.
alchemy, or what people thought of as alchemy at one time, the beginning of science, but
Yeah.
they were going off in an area that everybody thought was more like mystical.
See, we talked about the 1990s being the decade of DNA or molecular.
Right.
But
Yeah.
the 21st century, most people acknowledge the 21st century is going to be the molecular century. Because you're going to see things, we're not going to live that long.
Yeah.
I don't think I will, but anyway. But by, the year 2070, 2080, there's going to be things discovered genetically that are going to be so mind-blowing that they'll look at us and our little puny publications today.
We
and say,
were the
"Boy, these people-
Neanderthals.
Yeah, these people, they don't know anything about genetics. Yeah, the sad thing is, will they still be so ignorant in their adherence to evolution that they'll continue to just
Well, just-
deny the creator?
Just remember,
Yeah.
Mayans say we're out of here 2012, so.
So,
Before you get beat that long, don't worry about
Somebody asked me about that, and I just couldn't help but amuse and said, like, how long ago was that calendar originally written? Well, of course, it was written centuries ago. I said, well, maybe he just got tired of writing it, How many of our calendars do we take on for centuries on past? And I'm getting tired of writing this.
So we
thing.
said it just
I'm
kind
gonna
of
go take a
they ran out of
swim.
rooms.
Yeah.
And we're done.
so if you live to be 1,000 years, how much could you accomplish? I mean, pre-flood, how much did they know? How much knowledge was lost as a result of the flood?
Well, and again, modernistic man wants to view himself in such wonderful situation, yet we have to recognize that it wasn't until the 19th century that we actually surpassed Roman technology. We actually went down for a while and then came back up.
Well, that's like with Hugh Ross. He makes that crazy argument that the people in the antediluvian world would, if the universe really was billions of years old, they wouldn't understand it. And my argument is, I think they would have got it better than we do.
Yeah, in some ways, yeah.
Yeah, that's what I think.
Yeah. Well, we've talked about this before. What was the IQ of Adam and Eve? Well, I have no reason to believe, that their IQ would have been unmeasurable. We wouldn't have, it'd be 1,500. They would make us like dribbling idiots.
Yeah.
Well, Doug, that was pretty amazing.
Yeah, Kevin, I just get blown away with every time I hear him talk. We've interviewed him before out in Montana when
Yep,
we were out
Glacier
in the
National
National Park.
When he was talking about the genome, I tell you, Doug, when he talked about going to like King Tut's tomb and finding like another whole area out there, it just blew me away, Doug. And I'm still kind of processing some of that stuff.
Well, we'll see you next time on Revolution Against Evolution.